So we are recording this lecture as well. I'll post with all the other ones. All right, so here's where we're going to start, you guys. The endocrine system. We are going to go through most of these hormones. The only ones that we're not going to go through are the pineal gland. So you can actually X that out if you have that around. We won't be covering the pineal gland and melatonin. We actually won't be covering the thymus either. And then we also won't be going into detail on the gonads. Ah, darn. Okay, but I will say that there is an excellent class. Laura Morrow actually teaches a reproductive biology class in the animal science department. If you guys are interested in that, I highly recommend that you take her class. It's actually very, very interesting. All right, but the other ones we are going to go through, so let's go ahead and get started. I do want to talk a little bit about some of the ways that chemical signals can be delivered over short and long distances. If you need some text, this one summarizes it. But let's go through it panel by panel, okay? So here are the first two. One of the ways that you can deliver a chemical signal is through neurons. We've already talked about that. That was covered in exam 1. So a lot of those neurotransmitters are released from the axon terminal end. They diffuse across the synaptic cleft by diffusion alone because the distance between the axon terminal end and the postsynaptic cell is relatively small. So diffusion alone is very sufficient and very quick from the neuron to the postsynaptic cell. All right. However, if you're talking about signaling target cells that are very far away, right, a meter would take 32 years, which obviously isn't sufficient. It's not appropriate. So in that case, and this is what our lecture is really mainly about for the next couple days, there are non-neural endocrine cells. These endocrine cells release hormones into the blood supply, right? And then it travels through the circulatory system and finds their receptors on target cells. Okay, so these are non-neural endocrine cells. Endocrine cells, they release hormones into the circulatory system. All right, then I want to talk about, it's kind of a mixture between two systems. Neurosecretory cells. These are neurons that secrete molecules into the circulatory system. Neurosecretory cells. Now I want to give you some very important examples. Those molecules that are being released from the posterior pituitary, that includes vasopressin and oxytocin, they're released from neurosecretory cells. Okay, so this is an opportunity for me to kind of show you your hormone worksheet. You can actually see under posterior pituitary both vasopressin or AVP, arginine vasopressin, and oxytocin. So those are being released from neurosecretory cells. Yes?
Did you say the neurosecretory cells release?
It is a molecule. It's technically a hormone. Yeah. Yes?
What do they secrete molecules into?
Into the blood supply. Yep. And then they travel, vasopressin, as you guys know, travels to the kidney, right? To the epithelial cells that line the cortical collecting duct in the kidney. They insert water channels so that it allows an organism to reabsorb water and concentrate urine. So that was from exam 3, if you guys remember that. Oxytocin, on the other hand, is a molecule that is responsible for ejection of milk from the mammary glands. Ejection of milk. Okay, that's different from prolactin. Prolactin is, we're going to talk about prolactin that's secreted from the anterior pituitary. Prolactin is actually responsible for the production of milk. Okay, so there's a little bit of a difference. Oxytocin is responsible for the ejection of milk. And then I want to just introduce two terms, paracrine. Paracrine means, para always means next to. Paracrine means next to. Autocrine means self. Auto means self. So in this panel, panel D, this would be a typical paracrine signal. One cell type is releasing a molecule, a signaling molecule, which is traveling a short distance, it just relies on diffusion alone, to a different cell type that is expressing receptors for those molecules. And then it provokes a response, it stimulates a response. Okay, so that's paracrine, next to. Autocrine, like this cell on the right-hand side, is releasing molecules, and it's actually traveling right back and stimulating receptors on that same cell or cells of the exact same type. That's a typical autocrine signal. All right, so here's a nice table. You guys can kind of look at the differences between autocrine, nervous, and endocrine molecules, communication. It talks a little bit about secretory cells, target cells, the signal type, transport speed, duration. Okay? Let's focus a little bit more on the autocrine-paracrine side of things. Eicosanoids. These are a specific example of autocrine and paracrine cell signaling. You guys might have heard of prostaglandins. Prostaglandins are involved in pain reception. So when you actually take painkillers, like NSAIDs, non-steroidal anti-inflammatory drugs, or aspirin, those actually inhibit prostaglandins and their effects. So they're great painkillers. These are typical autocrine and paracrine signaling molecules. So in this case, arachidonic acid, you don't need to know this, but arachidonic acid actually goes through what's called a cyclooxygenase or a lipoxygenase pathways to produce prostaglandins, which is involved in pain, or leukotrienes. You might have seen commercials for allergies and asthma, right? Drugs that affect the lipoxygenase pathway help to reduce allergies and asthma. So, again, these are just typical examples of autocrine and paracrine signaling molecules. All right, so here's going back in more general terms. Let's talk about the difference between an exocrine gland and an endocrine gland. So exocrine glands, they actually secrete molecules, fluid and molecules. In this case, this is a typical acinar gland. We've talked about this many times before. Some of the examples that we've already used are salivary glands, pancreatic glands. Other examples are prostate and sweat glands. And the secretions actually occur in the acinus and then a lot of times are modified in the duct portion. And these secretions then end up in the external environment, which includes like the small intestines or Salivary glands, those secretions end up in the mouth. Respiratory surface, so a lot of times those secretions may end up into the lung itself or on the skin if we're talking about sweat glands. Other types of exocrine cell signaling are pheromones, allelochemicals. So pheromones, right, are scents that animals give off. So all of this is mediated through a typical exocrine cell signaling pathway. Now, endocrine glands, these cells, you can see it's surrounded by these capillaries. They secrete hormones or molecules that actually end up in the blood, the circulatory system, and then get transported throughout the body. Okay, so here's a general look at different types of hormones. We're going to start off by talking about peptide hormones and steroid hormones. Now, throughout the semester, we've kind of already talked about many different peptide hormones and steroid hormones. Now we're going to formalize it. So you might have already some information. This may be review for you. Then we'll talk about amines, specifically catecholamines and thyroid hormones, which are distinct from peptides and steroid hormones. So let's jump right into what peptide hormones are. A typical peptide hormone is like vasopressin. You guys already know what vasopressin is. Technically, vasopressin is called arginine vasopressin. Arginine vasopressin. So arginine is a typical amino acid. So these peptide hormones, they're actually synthesized in the rough ER, just like any other protein. And then they're stored in vesicles. They actually leave the cell by exocytosis. So once it's stimulated, these cells then, they fuse with the plasma membrane and then deliver the hormone into the circulatory system. So these hormones are usually soluble in aqueous solution, and then they travel through the circulatory system to the target cell, always dissolved in extracellular fluid. So the main idea here is, unlike steroid hormones, peptide hormones are hydrophilic, so they can't cross the plasma membrane easily. And their receptors are usually expressed on the plasma membrane, right? They're usually transmembrane proteins, receptors, where the hormone actually binds to those receptors, and there's an immediate effect, like vasopressin. If you guys remember from the kidney section, vasopressin binds to its receptors, and then it promotes those vesicles to fuse with the plasma membrane, inserting more water channels. So it's really rapid. It's like minutes. Very rapid effect. All mediated through the GS pathway. If you guys remember that. Increase in cyclic AMP, increase in protein kinase A, and then it promotes those vesicles to fuse with the apical membrane, inserting those water channels. All right, so here's something also that's very interesting in peptide hormones. Another very important peptide hormone is insulin. Insulin is actually a peptide hormone. So in this, I'll give you an orientation of this diagram here. On the left-hand side, this is the rough ER. This is where a lot of the protein synthesis occurs. So the ribosomes are kind of lined up here. You can actually see in this case, this ribosome is making messenger RNA. It's actually being delivered into the ER, endoplasmic reticulum, where it's then transported to the Golgi, right? Then it's packaged in these secretory vesicles, just waiting to fuse with the plasma membrane, right? A lot of times these peptide hormones then are delivered into the extracellular fluid. All right, but what you might not know yet is that the mRNA, it actually, I should say, translates, right? The mRNA translates, or the proteins are translated from the mRNA into these polypeptides, and they are actually in the form of pre-pro hormones. Pre-pro hormones. It's kind of interesting that they are first cleaved in the ER, and they're packaged in these vesicles as prohormones. So they start off as pre-pro hormones. They're actually, you can see the cleaved pre-pro hormone becomes a prohormone. The prohormone then actually is transported to the Golgi. And it's not until this last secretory vesicle does this prohormone then be cleaved into the active hormone. So there's a series of steps, cleavages along the way. Starts off as pre-pro hormone, and this is a good test question. Then it's cleaved into a prohormone in the ER, and then the prohormone is actually cleaved in these secretory vesicles to the active hormone. So let's take a look at this. This is just, example is insulin, right? So you can actually see that insulin is actually, let's go to this next one, you can see it a little bit better. Insulin is made as a pre-pro hormone. And then in the secretory vesicle, it goes from pre-pro hormone gets cleaved in the ER. And then it goes from the pro hormone, it's cleaved in these secretory vesicles. This A and B chain, this is insulin right here. You can see it's bound together by these disulfide bonds. And then the C peptide is just cleaved away from the insulin. So this might be easier to see. Pre-pro insulin looks like this. And then after 2 cleavage steps, you can see this is the insulin in the green and the purple, and then a free C peptide. Vasopressin is also the same way. You can actually see it's first synthesized as pre-pro vasopressin. Then the signaling peptide is cleaved off in the ER, endoplasmic reticulum. This would be the pro-vasopressin. And then in that secretory vesicle, it's actually cleaved again to arginine vasopressin, neurophysin, and a glycoprotein. You don't need to memorize neurophysin or glycoprotein. Just know where these steps, where the cleavages occur. Pre-pro insulin and pre-pro vasopressin are first cleaved in the rough ER. And then pro-vasopressin is cleaved again in the secretory vesicle. Pretty good, you guys? It seems complicated, but there is a pattern here for many of these peptide hormones. All right, so let's go ahead and move on to steroid hormones. Steroid hormones are not hydrophilic, but they are hydrophobic. They can easily cross the plasma membrane without any kind of protein involved, just by passive diffusion. They can migrate right across the plasma membrane. And because of that, they can't be stored within vesicles. They can't be stored within the cell. Because again, they can just move freely across vesicle membranes and plasma membranes. So usually steroid hormones are synthesized on demand. The receptor is usually in the nucleus. And once that stimulator, once the cell is stimulated, it actually starts to synthesize proteins within the nucleus. So it's synthesized on demand, transported to target cells by carrier proteins, and it usually takes about 24 hours before you see the effect of a hormone because protein synthesis is involved. So steroid hormones takes about 24 hours before you can actually see the results. Now, as you can see here, many steroid hormones are derived from cholesterol, right? I'll blow this up. You can see cholesterol can be cleaved to aldosterone, cortisol, testosterone, estradiol, which is estrogen. Okay, so there's a lot of steroid hormones are derived from cholesterol. You don't need to know the structure or memorize any of that. Just know that many of these steroid hormones come from cholesterol. Okay, so there's 3 different classes of steroid hormones. Number 1 is mineralocorticoids. This is aldosterone. You guys already know what aldosterone does. It promotes sodium reabsorption, and potassium secretion in the kidney. So it has effects on electrolyte balance. We're also going to be talking about glucocorticoids today. These are the stress hormones like cortisol. And then another type of steroid hormone are the reproductive hormones, estrogen, progesterone, and testosterone. They regulate sex-specific characteristics and reproduction. Okay, so briefly too, let's talk about the amines. We talked about peptide hormones, steroid hormones, and now the amines. Most of the amines are biogenic. They actually have, some of the examples, I should say, is acetylcholine, dopamine, epinephrine, serotonin, melatonin, histamine, and thyroid hormones. So they all possess this amine group. And as you'll see throughout this lecture, you'll see a lot of these amines have very diverse effects. And we're going to go into much more detail on the thyroid hormones. This is a cascade that you'll learn when we get to the thyroid gland, how T3 and T4 are produced. These are all amine hormones. All right, now as you go through too, I want to make sure that you guys are thinking about one of the major themes in physiology, those feedback control systems. So don't forget about positive and negative feedback controls. We're going to be talking about this throughout this lecture. A positive feedback control mechanism is just like oxytocin. So I talked about oxytocin in terms of milk ejection, but it's also very important in labor. So oxytocin is one of the classic positive feedback mechanisms. And in this case, it disturbs homeostasis. It just keeps getting, it amplifies up. The output actually acts as a further stimulus until something major happens, like birth. Okay, so for example, just oxytocin works like this. I think I talked about this on the very first day when I was talking about positive feedback mechanisms. Oxytocin is released when a woman goes into labor and there's stretch of the cervix and uterine contractions. So it sends a signal to the posterior pituitary to release more oxytocin, which increases the number of contractions, dilates the cervix even further, and that actually signals back to produce more oxytocin. So it amplifies the effect until a major event like birth happens. So in this case, it doesn't maintain homeostasis. It actually disturbs homeostasis. It amplifies it up. The output acts as a further stimulus. Negative feedback mechanisms, these are self-regulating. They help to maintain homeostasis, and you'll see many, many, many examples of negative feedback regulation going forward. All right, so let's go ahead and get started. Let's start with the pituitary gland. I call this the master gland. It's the Cadillac of all glands, okay? It's actually the major gland within your body, the pituitary gland. It is broken up into the anterior pituitary. So if you actually take a look at the brain here, this is the front part, the cerebrum. This is the back part. You can actually see the cerebellum here. And you can actually see the pituitary gland right underneath the hypothalamus. Okay, so the one in the front closest to your face is the anterior pituitary. The one in the back is called the posterior pituitary. Now, as I said before, remember vasopressin and oxytocin are actually released from neurosecretory cells. Take a look at these green structures here. These are the long neurosecretory cells that extend into the posterior pituitary. You don't see that with the anterior pituitary at all. You only see it with the posterior pituitary. So vasopressin and oxytocin are made in the hypothalamus. There's the supraoptic nuclei and the paraventricular nuclei. Vasopressin and oxytocin, they're actually made in these neurosecretory cells. Then they're transported along the axons to the axon terminal end, where they're then released into the circulatory system. So these are neurosecretory cells. The anterior pituitary is a little bit different. You can actually see the nuclei. They send, the molecules are released into these capillaries called the median eminence, where they then are transported by the blood supply, the hypothalamo-pituitary portal vessels, into the anterior pituitary, where they then signal these are different cell types that deliver molecules into the circulatory system and beyond. So it's kind of a two-step process. One hormone is released from the hypothalamus. It then travels by the blood supply to other cell types that release a different hormone. That's why when you take a look at your worksheet here, you'll actually see under the anterior pituitary, one hormone coming from the hypothalamus triggers the other cells in the anterior pituitary to release a different hormone. Okay, so that's very different from the posterior pituitary. Okay, so let's go into more detail. This is just review. This is how the posterior pituitary is organized. You can see those long neurosecretory cells. The opposite is the anterior pituitary, right? Delivers that first hormone into these portal vessels, hypophyseal portal vessels, into the anterior pituitary, where these cell types release a different hormone. Okay, so first let's concentrate on the anterior pituitary. There's actually 6 different hormones that I'd like you guys to know. You can actually see adrenocorticotropic hormone, ACTH. This travels to the adrenal gland to release glucocorticoids like cortisol, corticosteroids. We'll get into more detail on that in just a little bit. There's another one called thyroid stimulating hormone. Travels to the thyroid glands to release thyroid hormones. Okay. Growth hormone. Just know that growth hormone, its effects are mediated through insulin-like growth factors. If you block the receptors for insulin-like growth factors, you actually block growth hormone. We'll be talking about some of the sex hormones. These are released from the anterior pituitary, luteinizing hormone and follicle stimulating hormone. They do different things in males and females, and we'll get into more detail on that. And then, of course, prolactin. Now, we won't be talking about melanocyte stimulating hormone. It actually stimulates in some animals skin darkening, melanin. In frogs, especially frogs, reptiles, some birds. Oh, not birds, fish, sorry. Except birds. They have feathers. Okay, but we won't be talking about MSH's melanocyte stimulating hormone. Okay, so let's go through this. I'll talk about each one of these, and you can actually start filling out that chart as we're going along. Hi. Oh, I think it's over there. Okay. So, first of all, one of the hormones that's released from the hypothalamus is called TRH, or thyrotropin releasing hormone, TRH, thyrotropin releasing hormone from the hypothalamus, and it promotes the release of TSH from the anterior pituitary gland. Okay, so one thing that I've noticed, you guys, this may help you too. Tropin, a lot of the hormones that are released from the hypothalamus have a tropin at the end. If they're released from the anterior pituitary, they have a tropic at the end. Okay, so thyrotropin releasing hormone, or TRH, is released from the hypothalamus, travels down those portal vessels to the anterior pituitary to release TSH, which is thyroid stimulating hormone. Then thyroid stimulating hormone travels through the circulatory system to the thyroid gland, where it triggers the release of thyroid hormones. So some of the diseases associated with this, if there's some kind of problem with the secretion of either thyrotropin releasing hormone or thyroid stimulating hormone, or the production of T3 and T4, those are the thyroid hormones, it causes diseases like hypothyroidism or hyperthyroidism. Hypothyroidism is when your thyroid isn't working properly for some reason and you're not producing these thyroid hormones. A lot of people may gain weight, they may feel cold all the time. On the flip side, hyperthyroidism, your metabolism is very high, you might have a lot of energy, and you might lose a lot of weight. Now, it's not always this way. There's some inconsistencies. Some people that have hyperthyroidism may have problems gaining weight. So that's kind of a generalization. But we'll talk more about how it affects metabolism in just a second. Adrenocorticotropic hormone. It's actually corticotropin releasing hormone, or CRH, that's secreted in the hypothalamus, from the hypothalamus, travels down to the anterior pituitary, where it releases adrenocorticotropic hormone, travels through the circulatory system to the adrenal gland, where it releases glucocorticoids like cortisol, corticosteroids. All right, the next one, gonadotropin releasing hormone, GnRH. Gonadotropin releasing hormone is released from the hypothalamus, Again, travels to the anterior pituitary, where it stimulates the release of follicle-stimulating hormone and luteinizing hormone. So, in females, obviously it's kind of easy to remember, it stimulates follicle development and estrogen secretion. In males, it does something a little bit different. Travels to the testes, where it promotes spermatogenesis. Genesis is the production. Spermatogenesis from the Sertoli cells. So if you just remember that for females, the target is the ovaries. For males, the target is testes. All right, so luteinizing hormone is also released from the anterior pituitary following stimulation of gonadotropin-releasing hormone from the hypothalamus. Luteinizing hormone, there's a huge spike of luteinizing hormone right before ovulation in women. So there's this huge spike of LH right before ovulation. Causes ovulation and progestin production in females. In males, it again travels to the testes, signaling Leydig cells to produce testosterone. So again, the target is ovaries in women and testes in males. Okay, so moving along, you guys, prolactin actually stimulates the development of mammary glands and milk production. And growth hormone is the last one here. We're going to go into more detail on growth hormone. Growth hormone is responsible for cell growth and replication all through insulin-like growth factors. So again, if you block the receptors for IGFs, insulin-like growth factors, you actually block the effect of growth hormone. Growth hormone is stimulated by growth hormone releasing hormone from the hypothalamus. That's not very original, growth hormone releasing hormone. And what I think is funny is growth hormone inhibitory hormone, GHIH, was later found to be somatostatin. Remember I told you when we were talking about the stomach and the parietal cells, somatostatin literally inhibits everything. So somatostatin also inhibits the release of growth hormone. Okay, so here's going into more depth on growth hormone. What I really want you to remember about growth hormone is it does the opposite of insulin. You know how insulin promotes the uptake of glucose into the cells? So it lowers blood glucose levels, right? Growth hormone does the exact opposite. It actually causes elevation of blood glucose. And it does that by stimulating what's called gluconeogenesis. It's the production of new glucose. And that happens in the liver. It blocks glucose uptake and actually promotes utilization of fatty acids as energy instead of glucose. So the whole idea here is that it increases blood glucose levels. And what it does is it actually stimulates protein and RNA synthesis, promotes growth of tissue, cartilage, and bone. And this is a key one too. It actually works synergistically with thyroid hormones. So thyroid hormones don't work without growth hormone. So on the left-hand side, this is still the same, but this is a nice diagram that shows you some of the negative feedback mechanism, right? So growth hormone releasing hormone stimulates growth hormone from the anterior pituitary, which stimulates the liver to produce IGF. Once you have circulating IGF levels, it feeds back to actually inhibit the release of growth hormone and stimulates somatostatin, which inhibits growth hormone as well. Ghrelin actually has an effect on the release, stimulates the release of growth hormone. Now, what are some of the diseases? Pituitary dwarfism. This doesn't happen anymore, actually. Really quickly, a lack of growth hormone causes pituitary dwarfism. So with dwarfism, you can see that these small people are actually proportional. They just look very juvenile in appearance. And nowadays, when there's a lack of growth hormone, we just treat people, we give them growth hormone in their IV. So you can treat people with growth hormone if you suspect or if you diagnose them with pituitary dwarfism. This one is the opposite. You guys know Andre the Giant? I actually noticed that there was a show on HBO just recently on Andre the Giant. He actually suffered from an overproduction of growth hormone. This was called gigantism. He had a pituitary tumor that caused him to be over 7 feet tall. Now, a lot of times, these individuals don't live past 50 because their heart actually can't take their giant size. They have some heart issues associated with gigantism. So there is overproduction of growth hormone, thickening of the cranium and mandible, and premature death. There is a difference between acromegaly and gigantism. Gigantism happens during puberty. So they have very large size. Acromegaly actually happens after puberty, and you can see it's just a thickening. This little girl didn't have a pituitary disorder when she was little. It wasn't until after adolescence that she developed acromegaly because of a pituitary tumor. And you can see the thickening of the mandible and the cranium as a result. So acromegaly is after adolescence. Gigantism happens during puberty. All right, you guys, I will see you on Wednesday.
Did you guys share that?
No, we didn't. So I'm going to post that online so that you can get the answers. Just turn that in, yeah, with your section, and that's perfectly fine.
Okay.
Yep. Hi.
Having trouble logging in.
Oh.
Oh. That's weird.
It's Julia? Yeah. I don't know. Julia? She's having trouble logging into that. It says you need permission.
Good. How about you? I found that at the bottom of the pile. So.
Perfect. Yes,
Thank you.
Thank you.
So I was planning on coming and looking over my test during office hours.
Sure.
I also have, we talked a little bit about a couple of assignments that I missed.
Okay.
I was planning on emailing them to you, but I had already printed them and written them out.
Okay.
And I cannot figure out how to scan for the
Oh, that's
life
okay.
of me.
You can take a picture with your iPhone, or you can just give it to me.
Okay,
Yeah.
and I have them. So
Oh, okay.
When I come in
Okay, that sounds great.
and look over this.
Okay, thank you.
I'm just gonna have a logistics question, I suppose, about grades. So, I think I can get an A in this class because of I got a 91 in the first exam,
Okay.
85 in the second.
Yeah.
78 on the third, but I'm retaking the third.
Okay.
I know I just didn't have enough time, but I know I can
knock that out.
get that out
Yeah.
and everything. So
Yeah.
then, do you think it would be like possible for me to get an A in the class, like depending on how I do?
I
on
think
the...
I think you're definitely in the range.
Okay.
Especially when you have all those buffer points. Make sure you have all your homework and top hat in.
Okay.
Yeah, and your video project.
Oh right. Okay.
Yeah.
I forgot about the video project. So I was
Yeah.
looking and I'm like,
Yeah, you're doing really well. Yeah.
thanks. And then is there any way to make up the top hat, or are all the top hat entered already? Because there's
Yeah, we
something
could.
missing.
Yeah, so, there's a bunch on the back end that I'm
Okay.
still missing.
Okay,
So
I'm
let
on
me
the
get
back
those in.
end. Okay.
Yeah, yes.
Okay. So
Just, I'll probably have those in really soon.
Okay.
So, yeah.
perfect.
And then we can talk again.
Okay, yes, thank you so
Yes, you're welcome.
Hi, we just had a question about feedback on our.
Oh, sure.
Like, how do we know when we're ready to get that?
When I said that if you haven't heard from us, then it was acceptable. So just keep doing what you're doing. Just keep going with what you're doing. Yeah. So whatever your ideas are, just go with it. Because I only contacted those people that have like really. Do you want to talk to me about it still? We can go through it. Okay. If you guys feel pretty good about it.
So if we didn't hear feedback, then we're just supposed to
Yeah, just
assume we're good?
Yeah. In fact,
Going?
didn't I talk to you about some of it? No.
You kind of had, but
Okay.
we weren't sure, like, you were,
Yeah. No, I made an
Specifics
announcement.
as far
Yeah,
as-
I made an announcement in class today that said if you don't hear from us, just go. Yeah, just go.
Okay.
Yeah, go with it. Yeah, good job, you guys. If you want to come in and talk to me about it, though, I'd be happy to give you. If you have questions, if you have specific questions.
Yeah. Okay.
Yeah. Thank you. Okay, sure.
So we have a group member that we have not heard from or
Okay.
seen the first day of class.
Sure, and that's exactly why there's an evaluation sheet at the end. So I give everybody in your group a score. Everybody gets the same score. And then I go through the peer evaluations, and if you have somebody that just never showed up, then those points get taken.
Okay, yeah. We do want to start.
I'm not even sure if he's in the class anymore.
What's his name?
Ryan
Literally.
Oh, yeah.
Lada
Okay.
or something like that.
Yeah, and
yeah,
it's
I
not
know
gonna be
that
a
is.
hard
So
one because
I
it's just gonna
will.
be plopping you the
In fact, I've
clips.
already talked to him,
I don't.
and so he's going to go off on his own. Yeah. So don't worry about Ryan.
Okay.
Yeah. Okay.
Sounds
Yep.
good.
Hey, I'm just wondering about the makeup tests.
Yeah.
Can they lower our score in any way, or
Not at all.
no? Okay,
No. So you are set with the scores that you have. It's a no risk situation. So you
Okay,
might as
cool.
well just take it.
yeah.
And then if you do worse, don't worry about it. Yeah, it doesn't matter.
Okay,
Yeah.
great. Thank
Yeah,
you.
you're welcome.
Hi. I kind of had a question about a similar thing on the test that I'm making up. You mentioned last week that we can come in and
Yes.
look over at
Yep.
and
Yeah.
like see what we did wrong and that
Yes,
kind of thing.
definitely.
I am retaking test 2, so when would a good time
Yeah,
be to
do
come
you
in?
have the lab? You
I
don't-
do have the
oh,
lab,
okay.
yeah.
So, I was thinking maybe I would camp out in one of the conference rooms upstairs during Thursday so that you guys all could come in before or after the lab.
Okay.
And I'd just be doing some administrative work up there. Maybe 4:30. I'll make an announcement and try to find a good conference room. And then I will have copies of the test itself and the keys.
Okay.
So that you guys can really kind of, if you want to come in with a group maybe that you're going
Okay.
to take it with, that's probably best because then you guys can sit there and talk about it and discuss the answers and understand why you got it wrong.
Okay.
Yeah. Does that sound okay?
Yeah, that's great.
Okay, great.
Okay,
Yeah,
thank you.
Yeah. Hi.
Oh yeah, I was just trying to remember my question. Is you mentioned the differences between anterior and posterior pituitary gland. Is it simply the release of hypothalamus hormones for the anterior to trigger the release of?
Yeah, you mean from the hypothalamus? Yes. So the hypothalamus triggers release from the anterior pituitary by delivering
Those release hormones.
Yeah, release hormones, and delivering it into that portal system, and then that travels to the anterior.
That's the difference between the posterior and anterior.
So
Because
the
I remember your
posterior has those long neurosecretory cells that releases vasopressin. Okay. I know.
So they got the neurosecretory type of hormone, and then the other ones have the. Is it also?
Endocrine cells.
Okay, endocrine.
Yeah, call them endocrine cells.
All right.
Yes.
Okay, thank you.
Yeah.
Okay, kind of a loaded question.
Sure.
So we're doing the endocrine system
Yeah.
for our group,
Okay.
and we keep cutting and cutting and cutting.
Sure.
And we're finding it very hard because we want to do the a woman's cycle.
Oh, that's great. Yeah.
And
Make it simple.
Okay.
I would say really simple. Okay, you know what? I have a really good story actually.
Okay.
What you might want to focus on is the transition between the first trimester and the second and third trimester. So you can talk about ovulation. That's even before pregnancy.
Yeah, we're just doing like the 28-day cycle.
Okay. So
And
take that off.
because there's so much
Yes,
hormones, and we're
I
just like, which
know.
ones?
So I
We
would
cut
say,
it to 7 minutes, but it's still like way too long.
yeah, I would say let's just focus then on. Let's just focus on. Oh boy.
Maybe just do the first half of the cycle or the last half.
Yes, I would say, or even just ovulation.
Okay.
So we're talking about the corpus luteum, right?
I thought it was luteum.
Luteum, yeah.
Is it?
Luteum.
Okay, I don't
I
want to say
call
it
it
the wrong.
luteum, yeah,
Luteum. Okay.
but luteum. Let's see.
Luteum.
I call it luteum. So you know what I do? Here's the cheating pronunciation. Corpus luteum. This is always fun. How to pronounce? You have to get to a good, reputable one because some of them are.
Sometimes it's wrong too.
Yeah.
Okay.
How to pronounce corpus luteum? It's gonna be loud.
That's okay.
Corpus luteum.
Corpus luteum.
Corpus luteum.
Corpus luteum. Okay. And
Thank you, Julia.